Okay, hi everybody. Somebody can let me know if you can hear me okay. Yep, and we're on YouTube. Got it. Okay, thanks everybody for joining me. I don't think I have any announcements except next week and Friday, actually Friday and Saturday, there's a live event at Joel Salatin's farm in Virginia, I think. And I last heard there was still some places open. I know there's a limit on how many people. So if anybody wants to go there, and please remember to come up and say hello. And I'm not sure if somebody can put a link in to how to contact them or sign up, but if you can, that would be great. Okay, I was thinking anyways, I was going to talk about the heart and following up from the podcast I did with Dr. Manel Ballister. And then I've actually read 10 or so articles that he wrote and had another chance to speak with him, not recorded, for about an hour, at which time I think I got some of it, not all of it. But I decided, because one of my listeners sent me a very interesting article, and unfortunately it was about viruses. So I thought, rather than talk about how the blood returns to the heart, which is an incredibly fascinating story, which has a peripheral relationship to the COVID phenomena, very peripheral. I thought I would go over this study that one of the listeners sent me, because it actually has a lot to do with what we're talking about. What we're talking about means viruses. So I'm going to try to do my screen share technique again. So let me see if that works. Okay, I think that works. Can everybody see this? I can't access the chat, but maybe I can. Yep, it works. Okay, I'm going to get rid of this. Okay, this is not the slide I want to show. Okay, so here's the context of this article. As hopefully everybody knows by now, the way that virologists, scientists, medical doctors, and anybody on the street that you ask, how do you know there's a virus? How do you find a virus, especially a new virus, and show that it causes disease? So that is the question of the hour. Let me just get back to this one. So most people think that the way you do it is because a lot of people get sick. We know that's not true. Then science people, medical doctors, think that you find the virus in a bunch of sick people, and then you isolate it from sick people. You show it under the electron microscope, and that's not true either. There is no case, as we all know by now, of any pathogenic virus being found in any bodily fluid or tissue from any sick person. Every virologist agrees with that. That's just not how it's done, period. Okay, so how do virologists do it? They find the cytopathic effect, which this is Stefan's study. So they see this and they say that is proof there's a virus. Now, we're not going to go over that because I've been over that numerous times. Once they have the cytopathic effect, in other words, once they take unpurified material from a sick person, inoculate that, quote, grow it on V1RUS cells, which are monkey kidney cells, with all the starvation and antibiotics and calf serum and all the rest. Then they show you a picture of the breakdown products, and they point and say, here is the virus. So that is the next step in the virologist's proof that a virus exists. And this happens every single time in a discussion with virologists. It happened in the COVID-19 myths. The virologist came on, pointed with his cursor to a picture of the virus, this particle, which I'll show you in a minute, and said, there's the virus, that's proof. And then they go on to do genetic tests, not on that particle. Please understand that. The tests that they do are not on that particle. They are on essentially the brew that comes from the breakdown of the monkey kidney tissue. So that's very clear. But this picture of the virus is a key component of the proof that the virus exists. Now, let's get to the slideshow. When I say the picture of the virus, I don't mean this picture. This is a picture of the SARS-CoV-2. This is a picture of the virus, but this is obviously not a real picture. This is a computer-generated image that I heard, I'm not sure this is true, was the image by a graphic design department somewhere in England. They won a prize sometime even before COVID started with coming up with the best graphic design picture of the virus. Nobody thinks this is the virus. This is a picture that a graphic design team made on their computer. The same with this. This is a computer-generated image of SARS-CoV-2. No virologist, no medical person would show you this picture and say this is proof of the virus. Because if you did, then this picture should prove to you that unicorns exist. It was probably made in the same way, although this actually looks like a painting. Or how about this one? If you accept the previous two pictures of SARS-CoV-2 as proof the virus exists, then by rights, you would accept these two pictures as proof that unicorns exist. And I hope nobody is crazy enough to do that. So what pictures am I talking about? Pictures that everybody has seen. There's the picture. And there, with the arrow, is proof. SARS-CoV-2, there it is. You see the membrane, you see the proper dimensions for what we're calling a virus, and then you see little black dots like this. Those are the spike proteins that informed the graphic designers that all of these coronaviruses, particularly SARS-CoV-2, have spike proteins, and that's eventually became the sequence that they inject you with. That's what we're told. It's this picture. This is the proof. When you see this picture in the results of your viroculture, then you can use the template, and I'll explain that a little bit later, for SARS-CoV-2 to sequence the virus and to create and to, quote, find the whole genome. First, you have to see this, or maybe this. Here it is. You've seen this from all kinds of people. You've seen it from Dr. Mikovits. You've seen it from the virologist who talked to us on the COVID myths. I've seen probably 100 to 200 people send me this exact picture and say, this is proof that the virus exists. Now, here's proof, this one, that the virus is budding off of the tissue. So that's proof that the viral theory is accurate. The viral theory being, you don't see any viruses in the person. And, well, let me rephrase that. There's no viruses, then the virus is attached to the cell membrane of the cells of the person. They do that with the spike protein hooking up with the ACE2 receptor. They inject their business, which is the genetic material, into the cell, reproduce themselves, bud off the cell membrane like this. This is the picture of that. And then you make thousands, millions of copies. Now, I would just caution you that the only part of that you see is this and this. Here is the virus. Here is proof that it's budding off of the cell membrane. That's it. The rest of the steps you cannot see. You can't make a video of this happening. There is no proof except this is produced. Therefore, all the rest of those steps must have happened. So let's take a look at this particle. What do we know about this particle? This is the study that was sent to me. The title of the paper is Appearances Can Be Deceiving. Viral-like inclusions in COVID-19 negative renal biopsies by electron microscopy. This was published in August 2020, so it's pretty recent, in a journal which I never heard of, but as a peer-reviewed conventional journal called Kidney 360. So it's all things to do with kidneys. And my guess is, although I don't know this for sure, that these authors probably were not nephrologists, not virologists, although I don't know that for sure, which is important because that means they don't have so much skin in the game. So now this is going to get a bit boring and tedious, I would admit, but I'm going to read some quotes from this article and then explain what they mean in common English. because this is key to understanding the nonsense that we're talking about. First quote, we have observed morphologically indistinguishable inclusions within podocytes and tubular epithelial cells, both in patients negative for coronavirus disease 2019, as well as in renal biopsies from the pre-COVID-19 era. In other words, those little particles that I just showed you with the black dots, they're saying they see particles that are indistinguishable, identical to those particles within podocytes, I'm not sure what those are, and essentially kidney cells. And they saw them in people who were negative for COVID, and they saw them in kidney tissue from before COVID actually started, which means how did it get there? Because this supposedly was, quote, jumped from bats to humans, or was engineered only in late 2019 or early 2020. So how did these identical particles get there in the pre-COVID era? Next thing, quote, we postulated that endogenous mimickers could be present that are morphologically indistinguishable from SARS-CoV-2 virions ultrastructurally. Endogenous mimickers is exactly what I've been talking about all along. These are identical looking particles that come not from any outside virus, but from you, or the monkey kidney cells, or the fetal calf serum. They are morphologically indistinguishable. In other words, they are identical. SARS-CoV-2 virion, a virion is just one virus, so they are identical. Next quote, viral-like inclusions consisting both of single vesicles with diameters between 50 and 139 nanometers, as well as packed groups within larger vesicles, were found in all 15 cases, either in podocytes, tubular epithelial, or vascular endothelial cells. In other words, they looked at 15 cases in their kidney laboratory. They did biopsies. They looked at some structure called podocytes or kidney tubular epithelia, or the blood vessels in the kidneys. And in all 15 of these cases, 100%, they saw identical particles as SARS-CoV-2, the same size, 50 to 139 nanometers. But there was no chance that any, quote, SARS-CoV-2 virus was there. But they saw it anyways. Okay, this next one is a mouthful. A number of potential natural mimickers that can generate intracellular groups of round vesicles mimicking SARS-CoV-2 virions could be listed. The most likely being endocytic vesicles and endosomal compartment components, such as microvesicular bodies containing exosomes, among others. Endocytosis leads to the formation of 60 to 120 nanometer vesicles, which is within the size range described for SARS-CoV-2, which is 60 to 140. These endocytic vesicles may be coated by different proteins, one of which most common being clathrin. The presence of coating proteins may be responsible for the presence of electron-dense areas surrounding these vesicles, giving the appearance of a viral corona. Let's go back to the previous slide. So they're trying to find out, so what are these little particles that are being mistaken for SARS-CoV-2? Well, they could be endocytic vesicles. That means little round things that the cell takes in. They could be endosomal compartment components, that's including exosomes. These are just different parts of the cell, little vesicles, little round bodies that are naturally formed in the cell and that increase when the cell is dying. They are the same size. They are the same identical morphology, meaning picture, as SARS-CoV-2. And most importantly, they have a protein called clathrin. And lo and behold, this protein makes it look like they have spike proteins. This protein stains with the electron microscope, giving the appearance of a viral corona. Who would have thought? In other words, that viral corona, that picture, there's no evidence that that isn't just clathrin proteins stained on normal components of dead and dying cells. Next, alternatively, the viral-like inclusions could represent microvesicular bodies containing exosomes before their release onto the cell surface. So remember that budding thing, which is given as, well, look, it's budding off the cell. Well, it turns out those might be microvesicular bodies containing exosomes that get released, i.e. bud off from the cell surface, and they are unfortunately being mistaken for viruses budding off from the cells. Now, the next one. This again could contribute to the increased number of cytoplasmic vesicles within podocytes in patients with COVID-19 who are proteinuric, and could lead to the mistaken assumption that these represent virions. Individual exosome size vary, but they are generally between 30 and 150 nanometers, which falls within the size range reported for SARS coronaviruses. The potential for confusion of coronavirus particles with normal cellular components was in fact highlighted in a detailed ultrastructural study by, yes, folks, the Centers for Disease Control and Prevention, otherwise known as the Center for Deceit and Corruption of the SARS-CoV, responsible for the 2003 SARS outbreak. So let's break this one down. So it turns out that when you're sick, in other words, you have protein in your urine, which is what nephrologists look for, lo and behold, just like we've been saying, if you're sick, you make more of these particles. That's because you're either dead or dying. The particles are the exact same size as the coronavirus from SARS 2003. And who knew about this in 2003 based on, quote, detailed ultrastructural studies? None other than the Center for Disease Control knew that these images, which they were going to foist on us and are being foisted on us from virologists and the perpetrators of the engineered virus myth, they are being unfortunately fingered as being proof or represented as proof of the SARS-CoV-2, or in this case, SARS-CoV-1, 2003. And the Center for Disease Control said, nope, those are probably just stuff, exosomes, microvesicles being released from dead and dying tissue. Then, final one, recognition of this pitfall of viral-like particles actually dates back to the 1970s. Well, it turns out it actually dates back to Ender's original paper in 1950, but never mind. When the potential for mistakenly assuming that normal cellular components such as phagocytic vacuoles, microvesicular bodies, or extracellular breakdown products could represent viral particles was emphasized after a proliferation of studies claiming to have found ultrastructural viral particles within different types of cancer cells and fluids. In other words, anytime a person is sick or any bodily fluid that you look at, you could find these identical particles. They're more when you're sick, more when you have protein in your urine. And we know about these since 1970. We knew they were coming from you, not from some pathogenic exogenous virus. But we're all friends here. Who's to quibble? A little harmless mistake never hurt anybody, except when you end up destroying the entire world, and using that as the rationale for taking this particle, finding an alleged piece of it, and making a vaccine, so-called, whose intention is to make people sick. The other reason I want to point this out is when you hear that the virus genome was sequenced. Now, here's a way to look at this. We think that what happens would be similar to if you had a chair. So you want to know, what is a chair made of? That's equivalent to the sequence, the genetic sequence of a virus. So in the normal world, you start with a chair, and then you take it apart piece by piece, hopefully, and then you can find out the sequence. In other words, what steps were used to make this chair. And the same goes with, if you have a human, you can find out, you can take the genetic material from the human and find out what it's made of. That's how we understand what something is made of, is we start with the thing, and then we sequence it or find the components from the thing. But virologists don't do it like that. They take the pile of wood and nails and string, or whatever it is you make a chair out of, and they try to piece that together to make the chair. Now, think about that. If I said, here's a pile of wood, nails, can't remember what the name, the weaving material, like the willow bark or whatever you're going to make. Here's a pile, make the chair. And your question would be, which chair? Well, I'm not going to tell you which chair. You have to make the chair. So obviously you can't do that. You can only make the chair if you know which chair you're looking for. And since nobody has had the chair and nobody has a picture of the chair, they can't make the chair. So this picture came to the rescue. In other words, well, there's the picture. So we know it's a SARS-CoV-2. We know that somebody has sequenced the SARS-CoV-2. Therefore, that becomes the picture. That becomes what they call the template that we can create from that pile, the new chair. If you didn't have that picture, you couldn't obviously make the chair. So that picture that I showed you, which turns out to be a fraud, is the linchpin for using the SARS-CoV-2 as the template, which the next experiment is compared to. And lo and behold, they find the genome of the next one in their brew. Because they put the template of SARS-CoV-2, they put the picture of that chair, and they got that from the picture. And you take away the picture, there's no template, there's no genome, there's no variants, there's no engineered virus. And what in the heck is this all about? So hopefully that you got all that, and I'm going to take this off. And there we go. So let me just center this. With that, let's turn to the questions, and maybe I'll have some answers. Somebody said, so the question, are you familiar with the term magnetofection in the context of the mRNA gene therapies? If so, do you think that seems to be what's happening with people showing magnetic skin areas who've received the Pfizer or Moderna jabs. If not, here's a link to the 2002 research paper titled Magnetofection, Enhancing and Targeting Gene Delivery by Magnetic Force in Vitro and in Vivo. Thank you for this. I saw a video that somebody sent me of a doctor in England who was hypothesizing that this is what's happening, and I will take a look at this paper and I will look into it. I think all I can say right now is I continue to believe that the biggest play here is a kind of competing view of how biology works. The old view, and I would even go so far as to say the eugenicist view, is that humans and all living things are completely materialistic entities, meaning not in the money sense, but we're only made of substance. That substance is made of atoms, and that's all there is to it. And the DNA controls who we are and what we do. The other view is that the DNA does nothing. In fact, the DNA is only certain sort of like strings that make up the chromosomes. And the chromosomes are helical in nature, just like our heart, and just like, you know, the universe, so to speak, and sunflower seeds and snails. And the chromosomes are function as if they're antenna, collecting the information from the world in the form of thoughts and feelings and light and sound and all the rest of it. They interface with water, and out of that interaction, new proteins are created without any DNA, RNA involved. I'm not saying that DNA or RNA isn't ever involved, but I'm saying that the hypothesis is the bulk of the proteins are made without any. The play here with COVID is to subvert that and essentially inject you with some sort of potentially magnetic sensing antenna device that will disconnect you from the information of the universe. And so your information will now come from the information that you're given from those who create the injections. And they will essentially force you to make the proteins that the people who make the injections want you to make, including a toxin called the spike protein. That's what I think is going on. And the magnetic part is essentially to circumvent the normal chromosomal antenna function, which is based on the helical form, and to give you some other antenna, some other magnetic sensing device, which will hook you up instead of the sun and the moon to the man-made cloud. And all I can say is that's not a good idea. Okay. With Stefan's current study, why will the CPE genetic material only confirm for other RNA viruses? Why not DNA viruses? The CPE confirms for any virus, RNA or DNA. The reason we're looking at RNA is because the coronavirus and all the recent, AIDS, HIV, and Ebola, et cetera, are all RNA viruses. And that's the one that we're putting the yeast RNA so that we can synthesize the genome and prove that you can create the genome. That's like creating whatever chair you want if you have enough pieces of wood and nails and screws. But the CPE is not unique to RNA viruses. I don't really have any updates on shedding, except there's just more and more cases, which is very concerning. I think there's, I won't read the whole question, but there's a lot of things that are potentially toxic that are probably interacting with each other, like magnetic antenna. And so the magnetism is itself toxic. It's meant to get you to make a spike protein, which is toxic. The antenna forces you to be a collector of all kinds of electromagnetic fields, including 5G. So that's a toxin. And then there's lipid nanoparticles, synthetic motor oil. So that's a toxin. And PEG is a toxin, and a whole lot of the other things in there. And you put that together and you got problems. The CPE looks a little different depending on the culture and how you do it. The all-feared spike protein for the individual who had a mild illness, then quickly recovered, would the spike in that illness be the same as what's in the jab as far as harm to the bodily organs? Thank you. There is no spike protein attached to the SARS-CoV-2 because the SARS-CoV-2 doesn't exist. So in order for you to have gotten exposure to a spike protein, so-called naturally, you have to explain how the spike protein got in you. Now, it's possible it's in the water and in the air and in chemtrails and, there's possible a lot of things, but you have to remember all those things, it's technologically, logistically difficult to stabilize either an mRNA sequence or a spike protein. There's some idea that they put it in masks or they put it on swabs. All that's unproven. But if there was a spike protein, you have to explain how it got there, because it certainly didn't get in there through a virus, because there ain't no virus. Therefore, there was no spike protein. The spike protein, the virus is the cover story for the spike protein. Turpentine is used with cataracts and as well as everything else. Basically, I'm not sure how it's used. I think just orally. I don't think anybody ever put turpentine of any sort in their eyes, although someday we're going to have a podcast with Andy on nothing but turpentine, because he knows a lot more about it. So I think it was oral, and the purpose of turpentine is just to bind and excrete toxins. And that's how it works as a paint thinner, essentially. It's a universal solvent, so it binds with whatever impurity is in the paint, I think that's right, and flushes it away, and that's how it gets rid of it. So essentially does the same thing with the water in our body. Glaucoma is a longer story, so I'm going to hold off on that. Can you speak about the role of zinc with the immune system and any recommendations about taking zinc supplements? So we don't have an immune system, so there is no role of zinc with the immune system. But there are white blood cells, which are, one could call them, in a sense, an immune system. They're more, I would say, garbage collectors. So when you have debris, they are recruited to the area. Just like if you have a lot of garbage, you call an extra garbage truck and they hopefully take it away and recycle it. That's what white blood cells do, and they need zinc in order to do that. The best two sources of zinc are oysters, including our supplement called Oyster Max, which you can get from the Dr. Tom Cowan site, which is nothing but pure dehydrated oyster powder. The other good source of zinc is red meat. As far as I know, those are the two best food sources. and Oyster Max is the best supplement for that. The evidence that 5G causes symptoms, there are a number of papers written about this. I don't have them at my disposal right now. Maybe at some point, I mean, we put some sources in the contagion myth, so I would look there, and maybe someday somebody will compile a evidence that 5G causes harm, especially symptoms attributable to COVID-19. I'm hoping to do the heart thing next week, yes. Sorry, the next one is about ticks and Lyme, and I talked about that last week, so I would encourage you to watch that one. So in an autoimmune condition such as Graves' disease, that's hyperthyroidism, should we worry about how to decrease antibodies? Do you still think LDN is a good idea to improve an autoimmune condition? It's an interesting thing. The first thing, whenever a patient asks me, should I worry about such and such, I always say no, because worrying, as far as I know, doesn't help anybody about anything. So the answer to should you worry about it is no. Now, let's rephrase that. Should you do anything to decrease antibodies? Decreasing antibodies as a goal is way too abstract. And that's one of the troubles with medicine and in sense in life, is you think you want to be a better person. Or some people maybe think they want to be a worse person. Or some people think they want to do a lot of things. People say they want, I won't get into it. Let's just stick with that. They want to be a better person. The problem is, if you ask 100 people, what's the definition of a better person, they probably give you 100 different answers. And so there is no connection with that concept with your mind. And so the inevitable outcome is you will fail, which means you should, as far as possible, never do anything that the outcome will be guaranteed failure, because you might as well not bother. And so anytime you're trying to do something abstract, and you will probably notice that politicians in particular love to talk about abstract ideas, because I think they know that those will never happen, and nobody will even know whether they did happen or not. So I wouldn't set out to decrease antibodies, because you have no connection with that. But you might want to try to improve your health, and then you might even want to test whether the antibodies go down. Now, I understand that I'm splitting hairs a little bit there, but I'm splitting hairs for a reason, because we want to align our mind with our actions. So, for instance, you might want to take a walk or be in the sun, or not eat gluten, or not eat grains, or take LDN, or lots of things. And then you see whether the antibody levels go down. Because the antibodies just tell you how many toxins are in your blood that need to be bound. If you try to just decrease the antibody levels without decreasing the toxin levels, obviously you would make yourself worse. So you don't want to do that. You want to make yourself better by doing a variety of things, or maybe one at a time. And that's what I've seen with LDN. It improves the health of the people. That's low dose naltrexone. And then in many cases, the antibodies go down, but there's no evidence that it somehow makes your antibody levels go down. In fact, I would say that's the whole trick and principle of medicine. We try to get rid of the, quote, bacteria in a, quote, infection, but really the bacteria are there to clean up the debris. So what you really want to, the way you really want to think is you clean up the debris and then the bacteria leave. Now, it's true that you could measure the level of bacteria, you could measure the level of antibodies, and that might tell you whether whatever intervention you did actually worked. But a direct strategy to decrease antibodies or stop your nose from running is guaranteed to make you worse, because those two things are ways that your body deals with problems. You don't want to stop your body from dealing with problems. You only want to test to see whether your body has less problems to deal with. Is the IgG test meaningful? I assume you mean for coronavirus. The answer is no. What reason or excuse do virologists give for not isolating and gene sequencing what they claim are the viruses present after the cytopathic effect in the tissue cultures? Their excuses for not doing this earlier from the samples would not seem to apply. That's a good question. I haven't actually asked or heard them say, why don't they take the tissue cultures and then purify, isolate those viruses and sequence just the genetic material for those viruses, isolated and purified even from the tissue culture. And as far as I know, that's never been done. I could be wrong about that, but as far as I know, that's never been done. Why they don't do that? I think they don't think they have to. That picture is the proof. Then you just, every piece of RNA must be there. So they align it. In other words, they reconstruct it from what's there. And that's how it's done. Where do they get the template if they have no virus in the first place? They got it, the SARS-CoV-2 original template from SARS-CoV-1. Now, you may ask, where did they get SARS-CoV-1? They got it in exactly the same way they got SARS-CoV-2. Then you could ask, where did they get the original SARS-CoV-1? They got that from the, I'll just make up a number, 1993 coronavirus. So where did they get that one from? Well, they sequenced it in more or less the same way. So where did they get the original one for that? That was from the 1998 or 1988 coronavirus. And where did they get that? Same way. Where did they get the original? At some point, somebody made it up, and that became the answer. Is there a relationship between the flu vaccines 2019 with the deaths in the first wave? Maybe. It's an interesting question. I have two twin 4-year-old grandchildren whose parents were told at birth they had the genetic marker for cystic fibrosis. As of 4.5 years later, they showed no signs of it at all. What is your take on having the genetic marker for CS? It's an interesting question. I think that you can have sequences that are being made at various times for various reasons that may be part of those strands, which are being called DNA, and they're being expressed because of some changes or some impulse from the world and from the water in the tissues and cells. If you change the water and you change the tissue, my guess is it will change either the way that that gene is expressed, or, and I can't prove this by any means, it will either make that genetic marker go away, or it will change it so that it has no impact on your situation. The evidence for this is complicated, but it's the only way you can explain why, if you take any, quote, genetic disease, which I don't really believe there are genetic diseases, I think there's expression diseases, and that has to do with the characteristic and structure of the water based on the information from the world that's being downloaded through the chromosomes. So there may be abnormalities in chromosomes that make them to be less functional antenna, and that could be a source of the trouble, or that the dysfunctional antenna could create abnormal forms of water, which then create the abnormal expression of proteins. But I think laying the blame on the DNA is not accurate. And what I was going to say is every so-called genetic disease, one person dies when they're 12 and the next person lives until they're 50 and has a normal life, yet they have the same genetic disease. That cannot be if everything was determined by genetics. The genes, better said, the proteins are made through a complex interaction of the information downloaded in the chromosomes, interacting with the water, interfacing with the amino acids in our cytoplasm, creating the proteins that give us life. What is making us sick? I've been explaining that a lot. There's all kinds of things: electromagnetic frequencies, pollution, mass, swabs up your nose, social isolation, bad food, glyphosate, maybe flu shots injecting people, maybe things being sprayed in the air. There's all kinds of things. To say that this is a virus is nonsense. I only have a few more and my voice is giving out. Let me see if there's people who maybe I don't recognize. Interesting comment. I'll just read it and maybe not comment. Consider that this experience is a physical manifestation of a split between light and light-dark realities. The physical choice is, quote, by your own hand. Example, by getting the injection is a choice to continue in the light-dark reality structure, obviously in a different place. So it becomes more and more clear that there are more than one truth depending on your higher self choice. You will find the truth that solidifies your choice in reality. Thanks for solidifying the light choice with brilliant factual truths. Thank you. I'm not sure I understand exactly, but I think it's really comes down to choosing whether it's as strictly black and white as light and dark. I mean, it mostly seems like that to me. It's a choice between life and a version of life which is not life. It's connecting you to a synthetic biology, to man-made frequencies, which is, in my view, the antithesis of life. Is there anything we can do to protect ourselves from those that got the injection if you didn't? I mean, I don't really have a good answer for that because I don't have any experience or facts or studies or people that I trust. Uh, and there's many factors, like it could be the lipid nanoparticle, it could be the stabilized mRNA sequence, it could be the spike protein somehow, it could be electromagnetic waves being emitted from one person to another. There's a whole lot of possibilities. I said to a friend today, for the electromagnetic fields, I would use the biogeometry, biosignatures and L9 dependent, like the one that I wear on this is the biosignature, and the L9 dependent. You face that to your heart, and I pretty much wear that all the time. Doesn't mean I did any good for me, but I like wearing it. And, if it's a protein, that's more normal detox things like saunas and sweating and vitamin C and maybe zeolite and shilajit and those kind of things. Okay, last one. Or two. Are you planning to create a European distribution for the products you sell? I'd say that's a question for Asher. I don't really know. Seems like a good idea. Can you explain the story behind the blood-brain barrier? Everybody talks about the blood-brain barrier. I'd love to see a picture of the barrier. In other words, here's the blood from your kidneys, right? Here's the blood vessel, the capillary, and there's the kidney cell. What do you see? There's no barrier. There's just the blood vessel. I think there's a little bit of an extracellular space, and then you see the cell or the tissue. Now you do that with a neuron in the brain, and you see the same thing. There's no barrier. So where's the barrier? Now you could say, well, you can't see it because of the way you prepared the slide. And that's possible. So in other words, you have no idea what the barrier is made of. I've never heard anybody say the barrier is made of cellophane or lipid nanoparticles or a lipid bilayer. As far as I know, nobody has extracted this barrier and shown what it looks like or what it's made of. And before I believe it exists, I want to see it and find out what it's made of. I think that the reason they invented this story, like a lot of these things, is the pharmaceutical companies had all these drugs and people said, well, I don't want to take aspirin, I don't want to take Tylenol because it might get into my brain and mess up my brain. So you obviously don't want that. So you make up a barrier and say, no, Tylenol doesn't cross the blood-brain barrier. It doesn't get into your brain. And so it's good. It only poisons your liver, which, oh, well, good. Now I can take it. What they mean by that, if it means anything, is certain drugs show up more in the brain than other drugs. Certain chemicals are found more in the brain than other chemicals. Now, I don't know about you, but that doesn't prove to me there's a barrier. There's all kind of reasons why certain chemicals dissolve in certain tissue more than others. There's differences in the water concentration, et cetera. That doesn't mean there's a barrier. That means there's something called biodistribution, which is the same reason, I guess, right now we don't sell to Europe because we don't have a distribution center in Europe. And you may not have a distribution center for Tylenol in the brain. That doesn't mean there's a barrier. And I'm not even sure that Tylenol doesn't get in the brain. I suspect they just made that up as well. All right. Thanks, everybody. And hopefully we can move on next week and I can talk a little bit about the heart. Thanks for joining me. Maybe I'll see some of you in Virginia on Friday, and hang in there and have a good week.